And I think we can move on to our last speaker. That will be Conrado Pedebos. Conrado will talk to us about the Hitchhiker Guide to the Periplasm, Unexpected Molecular Interactions of Antibiotics in E. coli. Are you there, Conrado?
Hello.
Yep.
I can't start my video for some reason. It says the host has stopped it. Okay, start my video. Can you see me now?
Okay, yep.
Cool. Can you see my screen?
Yeah.
Okay, great. Well, so I would like to thank the organizers for putting up this really nice webinar series. So I'm Conrado, as Aneda said, and well, I work with Professor Simon Khalid Research Group in University of Oxford now. So before I was in the University of Southampton since last month, so I just transferred. But the whole of his work was done while I was in the University of Southampton. And the title of my talk will be The Hitchhiker's Guide to the Periplasm, Unexpected Molecular Interactions of Antibiotics in E. coli. So just to give you a brief introduction, this is an image of the E. coli cell envelope. It's formed by three major structures. So the first one is the outer membrane, which is the most external structure, and it is composed in the outer leaflet by lipopolysaccharide, which is quite rigid, and it protects the bacteria from the hostile environment. Then we have the inner membrane, which separates the cytosol from the rest of the molecules. Between these two membranes, we have the periplasmic space. And in the middle of the periplasmic space, we have the cell wall, which is composed by peptidoglycan. And in this periplasmic space, we have a lot of different proteins. These proteins can bind to the cell wall. They can span the whole periplasmic space. They can be embedded both in the outer membrane and in the inner membrane. And we also have osmolytes here. So this region is densely packed. And there is a formation of non-specific clusters for a lot of times. And this is something that is really important to consider because this can affect, for example, protein structures. So in a normal in vitro experiment or when we're doing your traditional ND simulation, what you usually have is a protein that is bound to, that is inserted in a solution with water and ions, or maybe you got a buffer there, but not much more than that. But in real life, what you do have is a complex mixture with different proteins interacting with your protein of interest. You have other molecular crowders such as osmolytes, and all of this can produce a whole range of effects, which can in turn favor specific conformations that you only see on your on real life situations. Also, in the context of this work, we need to consider the antibiotics, and in this case, we are talking about polymyxin, more specifically polymyxin B1. So polymyxins were used a lot during the 60s and the 70s, but they are quite toxic, so they were left aside for some time. But more recently, they have been used for because of the rise of multi-drug resistant bacteria, and they are quite potent, but they are also, as I said, very toxic. So this is the structure of PMB1. It is composed by a number of amino acids. 5 of them are this different amino acid called diaminobutyric acid. It is positively charged, so the polymyxin has 5 positive charges, but it also has some hydrophobic bits. More specifically, I want to focus on the acyl chain that you have here, and this confers an aspect of a cyclic lipopeptide and a cationic surfactant for these molecules. And as I said, it has toxicity, so effects. So it is neurotoxic and nephrotoxic. In terms of how they act, you can see that there has been many different proposals for their mechanism of action, but none of them have, it's something that's still up to debate. Like it's not really, hasn't been determined, and this has been studied since at least the 70s, so it's quite, has been quite a long time. And all of these works usually focus on the two membranes, so in the outer membrane or the inner membrane, but none of them focus on how does polymyxin cross through the periplasmic space, and like how do they cross the outer membrane. Then they have to navigate through that dense mixture, and then eventually they will reach the inner membrane. So the goal of my work was to assess the mechanism of antibiotics crossing through the cell envelope, and more specifically, how do they negotiate their way to arrive at the inner membrane. So one way that we think was interesting to study this was by doing computational studies, and we used molecular dynamic simulations for this work. We built a complex and crowded computational model of the cell envelope, and this work was published earlier this year. So if anyone is interested in having a look, you can check that. So just to tell a summary of our simulated systems, we did one control simulation of polymyxin in water and ions only. Then we built increasingly complex versions of the cell envelope. First, we have one system with only one brown lipoprotein and just 8 polymyxins. Then we expanded this to a system with 4 brown lipoproteins, following by another system with 4 brown lipoproteins, lol A, lol B, tau, and OMP, which are all proteins that are quite abundant in E. coli. And finally, we have the most crowded system, which is similar to the previous one, but we also included a range of osmolytes to try to really mimic what would be a more realistic model of a biological system. So to do the simulation, we use the GROMAC simulation package. We used both GROMOS 54A7 and Charm 36 force fields. So Charm 36 was used for the smaller BLP system, the smaller version of the cell envelope. And we also have an outer membrane that follows the composition of a biological membrane. So in terms of proteins, the proteins were chosen, they are all a bit related somehow. So we have BLP, PAL, and OMP, which are important for structural integrity. BLP is the only protein that is covalently bound to the cell wall, while PAL and OMP also bind to the cell wall, but in a non-covalent way. Then we have lol A, which is a lipoprotein carrier. Lol A has a function of carrying BLP through all the periplasmic phase. So it gets BLP close to the inner membrane, carries it to the outer membrane where we have lol B, and it delivers BLP to lol B, and lol B will then incorporate BLP to the outer membrane. So this is what the final system that we built looks like. So water and ions are hidden here, so we can better see the system. This is the outer membrane. We have proteins in yellow. We have the cell wall in pink, the polymyxins in blue, and we have the osmolytes in purple. So first we looked at the self-interactions of PMB1, so the control system, and we saw that PMB1 can form micelles in solution. The most prevalent form of these micelles were in the form of a tetramer, and the tetramer correlates well with experimentally measured micelles of polymyxins, more specifically polymyxin E, which is quite similar to polymyxin B. And we also looked at the behavior of these monomers while forming the micelles, and as you can see, the monomers get together for quite some time. So the micelle is formed for around 400 to 500 nanoseconds. Then they separate, so it can do this whole separating and forming again movement. So we also looked at the translational diffusion of all the molecules involved in our systems, to see how crowding affects that. And what we saw for polymixins, for example, is that there was a 100 times reduction in the translational diffusion, also which correspond to the more crowded system being the one with the smallest value for translational diffusion. We also saw that water gets slower due to the crowding, and the same for protein. So they all follow the same trend of the more complex system has a more complex, more crowded system has the smaller diffusion. And when we compared the translational diffusion for proteins with experimentally determined diffusion for GFP in the periplasm, we see that they are in similar ranges, so in the range of 10 to the -8. So this is good because it gives us more confidence in our model. We also looked at how PMB1 was interacting with osmolytes, and we saw that it can form clusters with the osmolytes. In this case, we give two examples of clusters, one forming in the cell wall surface with 3 polymixins and a range of different osmolytes. And we also see clusters forming in the membrane surface interacting with the phospholipids. And the osmolytes that are most prevalent around the structure of polymixins are OPG and glycerol. So OPG is a glucan that has negative charge. So this is good to interact with PMB1, which has several positive charges. And glycerol provides many opportunities to do hydrogen bonding with the polymyxin molecule. So this one makes sense that these two molecules were the more prevalent. We also saw PMB1 changing interaction partners many times during our simulations. In this case, I'm talking specifically about proteins. So as you can see here, polymixin can change from PAL to BLP, from BLP to LOB, from PAL to LOLA, and from one BLP to another BLP. So this is kind of the idea of the hitchhiking. So it uses the protein somehow to move and to navigate this complex mixture and jump around. And what was quite interesting as well is that we wasn't expecting to see this, but when we thought about it, we thought that actually it makes a lot of sense. Is that PMB1 bound to the LOLA cavity, and as I said before, LOLA is a lipoprotein carrier, and PMB1 is a sort of a lipoprotein, it's a lipopeptide. So it makes sense that it could bind to the hydrophobic site of LOLA. And in fact, it did during our simulations. So we saw this the hydrophobic bits of PMB1 interacting with the inside of the hydrophobic site, while the positive charges were interacting with the negative charges from the entrance of the hydrophobic site of LOLA. We also saw PMB1 binding to LOB. So in this case, it was even more canonical. And you can make an analogy with other lipophilic ligands. So in this case, we saw PMB1 inserting its hydrophobic acyl chain inside the hydrophobic cavity of LOB, and the residues from polymixins that interacted with the residues of LOB were similar to other lipophilic ligands, which was quite interesting. We also saw this behavior, which we call the walking behavior, where polymixins were interacting with Brown's lipoprotein, and it could use Brown's lipoprotein as a ladder somehow. So when they were located, let's say, in the middle of the BLP structure, then after a while during your simulations, they eventually reached the outer membrane or the cell wall. This happened more than once. So this is also quite interesting, and it happened for the 2 force fields that we were using, so both for Gromos and Charm, and which give us more confidence on what we saw. Finally, we saw also PMB1 binding to the cell wall. We saw 2 different binding modes. One, which we call the inserted binding mode, where polymixins inserted between 2 of the glycan strands and brought them closer together. and the other one is the surface binding mode, where polymyxin just attached to the surface of the polymix of the cell wall and couldn't move much anymore. Unfortunately, no polymyxin crossed to the other side of the cell wall during our simulations, which is a bit frustrating, but it makes sense because the cell wall is quite negative, so it attracts a lot the polymyxins. So to conclude, this is one of the most complex and crowded bacterial periplasm models to date. We saw that the PMB1 movement can be highly impacted by the many binding partners it finds. Also, we saw the PMB1 can bind to lipoprotein carriers, and this is something that we are hypothesizing here, that it maybe hijacks the bacteria's own lipoprotein transport system. Well, this could be something to look for in the future, and we hope that the data from simulations may provide the basis for further development of new antibiotics. Just as a final perspective, we're trying now to expand what we did in this work. So in this work, we only simulated a portion of the cell envelope with the outer membrane and the cell wall, and this bit of the periplasm. And now we're looking to do the full cell envelope, just also a big portion of it, using also the inner membrane and more proteins, more osmolized. So this is what we're looking in the future. I'd like to thank the Khalid group, especially Professor Simon Khalid, and also Ian Smith, who helped me with the polymyxin parameters and the charm simulations. I'd like to thank everyone for listening. Thank you.
Thank you, Conrado. Time for questions. You can make it through the chat or put it in the question and answer. We have one here, Conrado, from Dr. Anton Watts.
Hi, I guess I'm just down the road from you. But my question is really about the stoichiometry of polymyxin membrane interactions. You have a really nice feature where you can alter the stoichiometry. I can see that by number of polymyxin molecules. And the literature is full of lots of binding data, either to bilayers or to membranes. Are you able to test any of that binding data by simulation? Because that would really give a power of how the stoichiometry of membrane polymyxin interactions works. And that would be very interesting to do. I don't know if it's ever been done before.
I don't remember if anyone done this kind of thing. I mean, there are some studies of polymyxins and their interaction with membranes,
Sure.
but they're usually not using like a very complex model. They're usually the ones I've seen, at least, they have been done with simpler membranes, not with the like a full structural outer membrane. And the one I know, which is a study from 2015 from our group, actually used coarse grain simulations, which is not very detailed.
No
No
problem with the coarse grain, I must admit. Yes, no, you need atomistic. No, it's just to test the data with a simulation. I mean, you know, to try and link the experimental data with a simulation, that's always very, very important.
Yeah, it is.
Yeah, and there's a huge data out there.
Yeah, this is something we're trying to look at. Also trying to understand the mechanism of action of polymyxins, because I haven't discussed this much on the work, but you can see that it's still up to debate on how it penetrates the outer membrane and also the inner membrane to do their actions. But yeah, no, I haven't done that, but it's something definitely to look for the future.
Interesting, you can't see anything passing through the membrane.
No, it's really hard. People have tried to see the polymyxins crossing the outer membrane, and it's just the problem is that LPS is like a concrete.
Yeah, right.
It's just too hard. It's going to take a long simulation to do that. Probably.
Logically, so you know, you must have to be able to understand that. Okay, thank you.
Thank you.
More questions? Okay. Do we have more questions there, Eduardo? I put this now and I can see.
No, I don't think so. I don't think so.
Okay. So if we don't have, I would like to thank everybody and hope to see you soon in the 20 UPAB Congress that we will start in 2 weeks.